Welcome to my YouTube channel. Today we are going to talk about liquid biopsy, in particular about cell-free DNA. Liquid biopsy was introduced as a new diagnostic concept almost a decade ago for the analysis of circulating tumor cells in the blood of cancer patients. Liquid biopsy was introduced as a new diagnostic concept almost a decade ago for the analysis of circulating tumor cells in the blood of cancer patients. So, what is liquid biopsy? The concept of liquid biopsy refers to the analysis of any tumor derived material which is detectable in the blood and other body fluids, such as circulating tumor DNA or circulating tumor cells. Outline of my talk would be under following headings. Just a brief introduction about why we have to read about cell-free DNA and what are the differences between tissue biopsy and liquid biopsy, and a brief history of cell-free DNA, origin of cell-free DNA and factors influencing it, applications of cell-free DNA, what are the challenges we face, and pre-analytical variables, various cell-free DNA platforms, and we'll conclude with a small take-home message. Coming to the introduction of the talk, we know that cell-free DNA can be used as a surrogate marker for cancer diagnosis, prognostication, and monitoring of response. However, before applying cell-free DNA into cancer diagnosis, prognostication, and monitoring of therapy, we need to understand optimization and standardization protocols for pre-analytical variables. We need to refine our current analytical strategies, and most importantly, we need to understand the origin of cell-free DNA, its physical properties and dynamics in the circulation. Let us see what are the differences between tissue biopsy and liquid biopsy. As you all know, tissue biopsy is an age-old gold standard for morphological diagnosis, and the same tissue can be used for various molecular applications like fish, NGS, etc. But the procedure is painful and risky, and it may not be feasible always to access the tumor, and it may not be representative of the entire tumor due to tumor heterogeneity. Whereas liquid biopsy is minimally invasive. As it is minimally invasive, we can take the peripheral blood sample of the cancer patient to understand the tumorigenesis, metastasis, and therapy resistance. We can also use this platform for detecting cancer at early stages, and we can also take serial samples for monitoring of cancer development, disease progression, and recurrence. We can also use this platform for tracking response or resistance during and after treatment to allow for adjustments in real time. So, real time monitoring of the patient as well as screening of large population and detection of cancer at early stages, all these are possible by liquid biopsy. Same thing is depicted in image format here. So at initial presentation we can go for tissue biopsy as well as we can go for liquid biopsy. But for monitoring we cannot take repeat biopsies from the patient. In that scenario we can take peripheral blood sample serially and we can monitor for the development of resistance or relapse in the patient. After relapse, however, if the tumor is reachable, if the biopsy is feasible, then you can also go for tissue biopsy. So we know What are the differences and advantages of tissue liquid biopsy over tissue biopsy? Now we are going to see the evolution of cell-free DNA. So for the first time, Mandel et al. Has described cell-free DNA in the human plasma in 1948. Leon et al. In 1977 has studied free DNA in the serum of cancer patients and in autoimmune disease like rheumatoid arthritis. In 1997, Lowe et al. Has described fetal DNA in maternal plasma in serum. In 1989, Strawn et al. Have described cell-free DNA in plasma of cancer patients. Sindrasky et al. Have described the presence of RAS mutations in stool sample of colon cancer patients in the year 1992. Takeda et al. Have detected the presence of KRAS mutations in sputum of lung cancer patients in the year 1993. Sorensen et al. Has described presence of mutated DNA in human blood in 1994. So for the first time, ACOG recommended non-invasive prenatal testing in high-risk pregnant women with the help of liquid biopsy in 2012. In 2016, FDA approved PCRSA for EGFR mutations using plasma in lung cancer patients. Whereas very recently, FDA has approved NGS assays for multiple therapeutic targets using plasma from solid tumor patients. So with the brief background in mind, let's see the origin of cell-free DNA and its characteristics. It's a busy slide, but if you can follow my cursor, first we'll talk about cellular origin. As you can see here, cell-free DNA can be derived from tumor cells as well as non-cancerous cells like muscle cells, hematopoietic cells, and the lining of gut, etc. Within the tumor also, there is something called tumor microenvironment, which is again composed of non-cancerous cells like fibroblast, dendritic cells, macrophages, B cells, T cells, NK cells, etc. So all these can release cell-free DNA into the circulation. Now we know the source of cell-free DNA, then how it is released into the circulation. There are mainly two pathways by which cell-free DNA is released into the circulation. The first mechanism is cell death and clearance, under which we have apoptosis, necrosis, pyroptosis, mitotic catastrophe, autophagy, phagocytosis and netosis. Among these, apoptosis contributes most of the cell-free DNA in the circulation. Even live cells can also release cell-free DNA into the circulation by a process called active release. So, micromolecular structures, micronucleation, microvesicle formation, all these are the mechanisms by which live cells release cell-free DNA into the circulation. So, why we have to know the various release mechanisms of cell-free DNA? Because everything has its unique signature left behind. Suppose, for example, apoptosis, where you can see the cell-free DNA fragments which are being released into the circulation will typically have a size of 160 base pairs approximately and multiples of 160 base pairs. This is because of the fact that apoptosis is a programmed cell death, so even enzymatic action for the degradation of nucleic acid occurs in a regular fashion. So enzymes cleave the nucleic acid after a nucleosome. So you know that nucleosome is an octamer with with a strand of DNA, right? So the size of nucleosome with the linker DNA corresponds to 166 base pairs. So if the enzyme is acting here after one nucleosome, the released cell-free DNA will have a size of 166 base pair. So this is typically called trimodal signature of apoptosis. That means the cell-free DNA which is released from apoptosis will have a size of 160 and multiples of 160. Whereas necrosis largely releases large cell-free DNA fragments which almost ranges from 10,000 base pairs. Whereas mitochondrial DNA, the size ranges from 40 to 300 base pairs. The size of fragments released from netosis is unknown, and size of cell-free DNA fragments released by macromolecular structures ranges from 1000 to 3000 base pairs, whereas size of extrachromosomal circular DNA is 30 to 20,000 base pairs, and size of exosomes is 150 to 6000 base pairs. So now we know the various release mechanisms. Then comes to DNA binding. So if the cell-free DNA is free in the circulation, plasma nucleases act on the cell-free DNA and they try to degrade it. So most of the time cell-free DNA tries to bind to serum proteins and extracellular vesicles. So there is dynamic binding and dissociation which is happening in the circulation, based on which the quantity of cell-free DNA present in the circulation depends on. So cellular binding and uptake is again pH and temperature dependent. So if it is not bound to serum proteins and if it is not bound to cell surface receptors, then it is phagocytized by macrophages which are present in the liver and spleen, as well as it will be acted upon by DNase 1 enzyme, and the leftover thing will be excreted in urine. So the half-life of cell-free DNA in the circulation will range from 16 minutes to 2.5 hours. So we have seen the source of cell-free DNA, various release mechanisms, DNA binding and uptake, and digestion and clearance in the circulation. So normally in a healthy individual, the cell-free DNA which is released into the circulation will be majorly by hematopoietic stem cells. So that will be cleared off by phagocytic mechanisms, as well as it will be excreted in urine. But when you have abnormal conditions in your body like cancerous state or autoimmune diseases or inflammatory conditions, even physiological conditions like pregnancy, the turnover of cell-free DNA is more. So our phagocytic system cannot uh, phagocytize all the cell-free DNA which are present in the circulation because it is exceeding the phagocytic capacity of our body. So in those situations we can see raised levels of cell-free DNA in our circulation, which can be used for further applications. So here cell-free DNA from healthy individuals is represented in this gray color, and low cell-free DNA cancers are depicted in this color, and high cell-free DNA cancers are depicted in light blue color. So if you can see here, healthy individuals will have approximately up to 10 nanograms. Again, it depends from person to person. Whereas in renal cancers, glioblastomas, and bladder cancers, pancreatic cancers, the secretion of cell-free DNA itself is very low. Whereas for cancers like melanoma, breast, ovarian cancers, lung cancers, colorectal cancers, the release of cell-free DNA into the circulation is more. So it also depends on the tumor type. For a given tumor, there are certain factors which affect release of cell-free DNA, like mitotic activity of the tumor, presence of necrosis, degree of vascularization, and the tumor size. So logically, the larger the tumor size, the more cell-free DNA which is released into the circulation. But again, it depends on the tumor type also, as we have seen in the previous slide. So there are patient factors and there are tumor factors which are affecting the quantity of cell-free DNA which you get from the plasma. You have to know about cell shed. Cell shed is a phenomena which tells about the rate of release of cell-free DNA by tumor cells and renal clearance, based on which quantity of cell-free DNA in the circulation depends on. More the renal clearance, lesser is the cell-free DNA available in the circulation. Lesser the renal clearance, more is the cell-free DNA which is available in the circulation. Let's see the quantity of cell-free DNA in healthy individual, in pregnancy, and as well as in cancer patients. First, let us talk about healthy individuals. So, in healthy individuals, the cell-free DNA concentration, as I said, it is mainly from hematopoietic stem cells, which releases genomic DNA into the circulation. So it ranges from 1 to 10 nanogram per ml in the plasma. Again, this varies from person to person based on the rate of clearance, phagocytic activity, apoptosis and necrosis which is happening in your body. Everything contributes to your concentration of cell-free DNA. So in pregnancy, it is around 150 nanogram per ml. Again, this is contributed by both fetal as well as maternal cell-free DNA. As the gestational age increases, the cell-free DNA concentration will also increase in pregnancy. Whereas in cancer patients, on an average, it, it is around 10 nanogram per ml. And this fraction of cell-free DNA is widely variable. As we have seen, it depends in turn on tumor type, disease stage, and patient factors. As the concentration of cell-free DNA is highly variable, it is depending on so many factors. Our commercial assay should have a sensitivity of 0.1%. That means should be capable of detecting one mutant molecule out of 1000 cell-free DNA molecules. So the size of cell-free DNA is normally ranges from 170 to multiples of 170 because of the fact that nucleosome along with the linker DNA is roughly measures around 170 base pairs. However, the quantity of lengths corresponding to 2 or 3 nucleosomes will be very less in our plasma. So most of the cell-free DNA will be around 170 base pairs. Next, coming to applications of cell-free DNA. So cell-free DNA can be used for screening, detection and early diagnosis of cancer, for cancer staging and prognostication, initial therapy stratification, that is selecting a targeted therapy, monitoring of response to therapy, monitoring acquired drug resistance, monitoring measurable residual disease and relapse. So all these indications we know, but there is a special indication where we can use a liquid biopsy, that is cell-free DNA profiling, that is cancer localization. Sometimes even IHC may not help in cancers. So in cases of carcinoma of unknown primary, based on the tissue specific nucleosome spacing and methylation patterns which varies from tissue to tissue, so based on the patterns we can localize the cancer, we can know the primary. That is one advantage of doing cell-free DNA in cases of carcinoma of unknown primary. So you can take single sample whenever you need, or you can do serial sample monitoring also to study the progression of disease in real time. We know the applications of cell-free DNA, but there are challenges which needs to be overcome for successful cell-free DNA tumor profiling. So what are the challenges? The cell-free DNA uh, quantity will be low and variable because of inconsistent tumor fraction, and subclonal genomic alterations are very difficult to detect, and we cannot differentiate errors during PCR amplification and NGS sequencing errors with the subclonal genomic alterations. So there are chances of false positive and false negative results. So mitigation strategies required for sensitive and accurate NGS results. So to overcome all these challenges, we need to follow some protocols for successful detection of true variants in the cancer patient. Let us see the pre-analytical variables. So starting from the collection tube to the cell-free DNA quantification, everything should be chosen very cautiously in order to minimize the errors. So there are options like we can collect serum directly, anticoagulated tubes can be used, cell stabilizing tubes can also be used. But the problem is we have to know whether it is causing genomic DNA contamination with the cell-free DNA, and anticoagulated tubes like heparin and citrate cause inhibition of PCR. So what is the storage time if you are not going to process it immediately? Is it 6 to 24 hours, 24 to 72 hours, more than 72 hours? Which one is ideal? Again, you have to consider the factors like if you are keeping the sample in a tube and you are leaving it like that for hours together, there are chances of blood cell lysis and releasing of genomic DNA into your plasma. So this will dilute your cell-free DNA which is released from the tumor cells. You will have reduced plasma volume yield, reduced cell-free DNA integrity because of increased fragmentation. As I said, plasma will have nucleases, so it will fragment your cell-free DNA. What is the storage temperature? Is it at room temperature or 2 to 8 or minus 20 or minus 80 or it is more than 30 degrees? Again, you have to consider all these factors. Plasma processing. Do you have to do only single centrifugation which may lead to incomplete separation of whole blood fractions, or double step centrifugation or triple step centrifugation? If you centrifuge it vigorously, it may cause mechanical lysis of leukocytes, again contaminating your cell-free DNA. So what should be the method of cell-free DNA extraction? Is it silica based, magnetic bead based or microfluidics? Again, you have to see the compatibility of the extraction method with the collection tube, and you have to know the cell-free DNA yield with each of these, and cell-free DNA fragmentation. You have to select cell-free DNA quantification methods. Is it spectroscopic, fluorometric, capillary electrophoresis, qPCR? Again, you have to consider things like accuracy, lower limit of quantification, what we are measuring exactly. Is it cell-free DNA or genomic DNA or both? So all these are the pre-analytical variables. So for specimen collection, we have two choices. Fixative containing blood collection tubes. that can be stored at at room temperature for 7 days, but it is expensive. So it can be used for shipping purposes. When you are expecting a delay in your processing, you can advise to collect the sample in fixative containing blood collection tubes. EDTA, if you collect blood in EDTA, then processing should be done within the first 6 hours of sample collection. It can remain stable up to 48 hours, but the chances of lysis of your blood cells and release of your genomic DNA into plasma and diluting your cell-free DNA is more. So this flow chart will tell you the proper guidelines for each step. The choice of matrix should be plasma, because in plasma you will have proteins, so your cell-free DNA will be bound to proteins, so it prevents uh, the degradation of cell-free DNA by nucleases which are present in your plasma. So plasma is the best matrix to choose. Then you have to process it immediately. You should not wait. You have to process immediately, as soon as possible, preferably in the first 6 hours itself. Then the choice of tube in which you have to collect sample. EDTA vacuitainer is best because fixative containing tube is ideal, but it is costly. So you can proceed with EDTA vacuitainer. Then centrifugation protocols. You have to follow two-step centrifugation protocols. First centrifugation step is at low speed followed by high speed. So all cellular elements will be cleared off in at slow speed centrifugation, and the remaining cellular debris will be cleared in high speed centrifugation. So after centrifugation, the collected plasma ideally should be utilized. If you are not going to isolate DNA from your plasma, then you can store it -80 degrees. Better to avoid frequent freeze-thaw cycles. So if you want to thaw it, you can keep it at room temperature only, because there are no guidelines for plasma thawing as such. Again, for your DNA isolation, automated extraction is most suitable. So based on the automated extraction platform, the protocol changes: protein digestion, buffer type, elution volume, elution regime. Then after isolating your cell-free DNA, storage of isolated DNA. So it can be stored in normal tube. No need to use any specialized DNA low binding or protein low binding tubes. So plasma is the choice of matrix. Immediately you have to collect your sample in EDTA K3 vacuitainer. You have to process it immediately. You have to follow two-step centrifugation protocol, slow speed followed by high speed. Then plasma should be stored at -80 degrees. Avoid freeze-thaw cycles. If you want to thaw it at room temperature. Follow automated extraction protocol whenever possible, and store it in normal tube if you want to store your isolated DNA. One more thing is that the primary matrix used for cell-free DNA analysis is blood plasma according to recommendation. However, you have to remember that other body fluids, if they are in close proximity to the tumor, like in lung cancer patient, pleural effusion, so better you take pleural fluid rather than taking a peripheral blood sample in that scenario. So what are all the cell-free DNA platforms which can be used? We can use real-time PCR It is less complex, low sample requirement, low analysis time, better analytical sensitivity. But the disadvantage is that we can only target limited number of targets. In case of digital droplet PCR also, there is same disadvantage. That is, limited number of targets can be studied. So to study multiple targets in the patient sample, we can either go for whole genome sequencing, exome sequencing, targeted sequencing, shallow whole exome sequencing. For targeted sequencing, the depth of coverage will be more and it is cost effective. That's why targeted sequencing is preferable in out of all NGS methods or platforms. So before using NGS as your platform, you have to know certain things. MAF stands for mutant allele fraction. So it is nothing but the ratio between amount of mutant allele present in your sample by wild type alleles, which generally correlates with the tumor burden. But mutant allele fraction is a measure of genetic heterogeneity, but it is dependent on the cells assessed. If you are taking only mutant cells, then it appears to be like more of mutant allele than wild type allele. If you are taking more of genomic DNA than the cell-free DNA from tumor, then it appears as if there is low mutation burden and more of wild type allele in your body. So based on the types of cells you are taking for analysis is important. So this slide shows various platforms and the detection limit of MAF. For quantitative PCR it is 10 to 20%. For cold PCR it is 0.1%. For digital PCR and beaming it is 0.001 to 0.05%. For genome wide sequencing methods it is 5 to 10%. So with the help of targeted sequencing to reduce our limit of detection to 0.1%, we need to minimize the background error rates of sequencing, as well as we have to increase the signal to noise ratio of samples. So this can be achieved by molecular barcoding, which involves the allocation of UMIs, that is unique molecular identifiers, to each DNA template, which should be followed by amplification of each uniquely tagged template to create UID families. Okay. So cancer or patient-specific multiplex panels can be used in conjunction with targeted sequencing methods to achieve a MAF of as low as 0.01% to 0.5%. Sampling noise is another major factor that limits detection sensitivity of your sample due to limited blood availability. In that scenario, you can take large volumes of plasma by plasmapheresis, and you can also yield cell-free DNA by implanting devices that selectively capture your cell-free DNA, which are not done routinely, but these are the methods where you can take more of plasma. If you take more volume of plasma, there are chances that you get more quantity of cell-free DNA. So what are PCR amplification errors? How they affect your limit of detection? So mutations can be introduced during PCR by DNA polymerase in misincorporations, or they can also arise from DNA damage, which will appear in only one of the two DNA strands, so they are not counted as real mutations. So DNA polymerase can also misincorporate some of the bases, so that leads to amplification error that appears as a mutation, but that is not a true mutation. Also, when you have damaged DNA because of maybe long storage or vigorous shaking, so if the DNA is degraded or damaged, so that thing will be visible on one of the two DNA strands. If miscorporation events by DNA polymerase occur in the first round of amplification, all these will be propagated to the daughter molecules and they are likely to be erroneously scored as mutations. If the error is occurring in the first round of PCR itself, then all the amplicons which we are getting from the first parent strand, will carry the same erroneous mutation, erroneous misincorporation of base or error which has happened in the parent strand will be visible in all the daughter strands. So that will be considered as mutation. Usually PCR amplification errors occur in the late cycles. However, because the same mutation is unlikely to occur in unrelated molecules, these artifactual variants would give the appearance of subclonal mutation. When sequenced, these subpopulations of related molecules will all contain a copy of the original misincorporation event and they will be erroneously scored as a mutation. So, why DNA polymerase is causing misinsertions? Because DNA polymerases typically uh, make misinsertions at a frequency ranging from 10 to the power 4 to 10 to the power 6. So, which can lead to thousands of false positives on a typical sequencing run. That's why we check the fidelity of DNA polymerases, that is proofreading activity, to check whether the correct base has been incorporated or not. Damaged or degraded DNA is particularly sensitive to this form of error because of the prevalence of DNA adducts that cause erroneous base pairings during polymerization. We can remove this DNA damage with the addition of glycosylases or in vitro repair kits, but not all mutagenic lesions are recognized by these enzymes, nor is the fidelity of repair perfect, so limiting their utility in error correction. So solution for reducing sequencing error and PCR amplification errors is using UMIs. What are UMIs? UMIs are also known as molecular barcodes or random barcodes. So why we use UMIs? It is useful in three scenarios particularly. When you are having very low input sample, when you are going for a deep sequencing of RNA libraries, and for the detection of ultra low frequency mutations in DNA sequencing, you will use UMIs. In addition to that, UMI analysis is an excellent QC tool of library complexity. So at what step we are using unique molecular identifiers in our sequencing? UMIs are incorporated in sequencing libraries before PCR amplification step. So before PCR amplification itself, you are tagging your sequence with UMI. So it will correct your PCR and sequencing errors so that it improves accuracy and quantification, and but we need to pay special attention in the bioinformatics pipeline. So these are the adapters, these are the indices which you are using for patient identification. Apart from that, you are using UMI, unique molecular identifier, for each molecule. So you can use single or duplex UMI for your sequencing. So duplex UMI, you are attaching UMI tag on both ends. Unique molecular identifiers will have a random sequence of bases. So if you have DNA molecules like this 1, 2, 3, so you are using different UMIs. This yellow color represents one UMI, blue color represents say UMI 2, red color represents UMI 3. So after sequencing, you will gather all the yellow UMIs at one place, blue UMIs at one place, and red UMIs at one place. Then you will check for the mutation if it is present in all the strands. If it is not present in all the strands, that means that is not a true mutation. That is maybe due to sequencing error or PCR amplification error. Let's see this example. This is original DNA fragment, okay, which is carrying a true mutation, which is there on both the strands. So you have sequenced it. When you sequence it, this particular mutation should be there on forward strand as well as on reverse strand, right? These red dots indicate the sequencing or PCR error. It's not a true mutation because it is there randomly on one strand, okay, it's not there in all the strands. So when you do a bioinformatics pipeline, when you run a bioinformatics pipeline for error correction with UMIs, only the true variant will be called as a true mutation. All these red dots, that is PCR errors, sequencing errors, errors introduced during the sequencing workflow, will be neglected. So ultimately you will be calling a true variant as true mutation in this patient. This is possible because you have already tagged your sequence with UMI. Only the sequence with UMI should be sequenced, and if it is sequenced properly, then the same mutation will carry forward. That's how we are taking this mutation as true mutation. Take take the other example. This is another patient sample's DNA, which is not having any mutation here, but when you sequence, you are getting mutations here, random calls. Here also you are getting. So when you just take uh, these two separate strands, that is forward strand separately, and see, then you tend to neglect these random calls here. And if you take only reverse strand and see, then you tend to take this call because it is appearing in all the reverse strands, so you'll take it. But when you combine and read this DNA, that is the forward strand and reverse strand, when the mutation occurs, it should be there on the forward strand and reverse strand also. So it is not there on the one of the strands, so it should be neglected. Whereas here in this scenario, it was there on both the strand, forward as well as reverse strand. So it was considered as a true mutation. So again, for better understanding, after PCR amplification, you don't know whether these are all or PCR duplicates or your true copies of your original DNA. So without UMIs, you cannot say, you can, okay, all these are your DNA. So you take by in silico analysis, all this will be reduced to one molecule. When you tag your sequence with UMI, then you can differentiate them. You can differentiate based on UMIs. So UMI 1, 2, 3. So these will be grouped according to the UMA signature. So this bundle is for one molecule, this bundle is for other molecule, this is for third molecule. So you are taking three different sequences here which are correct. So now we'll see with an example. So you had a mutation here and randomly mutation here. So without UMS, you tend to take this strand with mutation and this strand also with mutation, and some of the strands without mutation, you tend to take all these three. With UMIs, when you are checking for the presence of mutation in all the strands, this particular UMI, it carries mutation only on one strand. It is not there in rest of the strands. So it may not be a true mutation. It may be simple misincorporation by DNA polymerase, right? If it is a true mutation, it will be present on all the strands. So only this UMI has been considered as a true call. So we need to have a special protocol for UMI processing. After tagging with UMI, you need to deduplicate it also. So that will be done by your informatics pipeline. So after demultiplexing, you will create BAMs. Then after creating BAM files, you need to deduplicate all your duplicating errors in your PCR. So deduplication is nothing but removing your PCR duplicates with errors. Okay. So error correction happens after generating BAM files. So again you take all the read families together, combine both forward and reverse strand, then finally you will call that variant as a true variant. So to summarize, we have seen the pre-analytical optimization, like which tube to be collected, how to process it, what are the storage conditions, and proximal sample collection. For brain tumors, you can take CSF. For oral cancers, you can go for sputum. For colorectal cancers, you can take stool sample. For bladder cancers, you can take uterine samples. For ovarian cancers, you can take uterine lavage. Okay. Then size-based enrichment of tumor-derived cell-free DNA should be done because we need only shorter fragments, because apoptosis will release fragments of size ranging from 170 to 500, multiples of 170. So if you are going for ddPCR, you tend to separate your sample by partitioning. If you are going for targeted sequencing, you will amplify, and before amplification you have to attach your UMIs. Then you have to run a separate bioinformatic pipeline to identify your true variant. So parallel multi-analyte testing can also be done by using all these things from your liquid biopsy: miRNAs, proteins, cell-free proteins, exosomes, metabolites, methylation. All these can be combined and you can get an ultimate report for that patient with the help of liquid biopsy alone. So UMI based error correction improves sensitivity and accuracy of liquid biopsy testing. So you have to incorporate this strategy in your bioinformatics pipeline as well to reduce the impact of PCR and sequencing errors. You need to optimize additional parameters like DNA input, sequencing coverage for optimum efficiency. So to conclude, circulating cell-free DNA testing allows detection of a range of genomic alterations derived from circulating tumor DNA. Tumor type specific evidence needs to be evaluated to identify suitable clinical applications like genotyping, prognostication, targeted therapy, treatment resistance, immunotherapy, etc. Liquid biopsy genotyping results for metastatic cancers are highly concordant with the tissue biopsy results. So you can start doing liquid biopsy genotyping in your lab. A negative liquid biopsy result may be due to a variety of biological and technical factors as we have seen before. So in that scenario, you can request for a tissue biopsy to just confirm your true negativity or just to rule out your false negative result. Newer technical and clinical applications of circulating cell-free DNA tests continue to emerge, requiring ongoing assessment of clinical translational opportunities. We have seen that numerous pre-analytical factors contribute to recovery of cell-free DNA. So, cell-stabilizing tubes are becoming the standard for liquid biopsy applications. Even ISO recommends cell-stabilizing tubes. Specimen handling, storage, processing need to be optimized for clinical testing. DNA extraction methods will also influence your cell-free DNA and circulating tumor DNA in your sample. Pre-analytical enrichment could improve assay sensitivity.